the integumentary system, then we do bone, cartilage, muscle, nervous, and we kind of finish out, and then we roll into block 3 of neuro, which is my favorite. Well, one of my favorites. I like musculoskeletal better, but neuro is real interesting to me. And hopefully, the goal, unlike other people would tell me, is not to make you, or not to show you how much I know about neuro, it's to, I'm going to give an example. The gentleman who taught this course before me had a PhD like in genetics. So guess how big the genetics section was when we went through it. So you can imagine, I cut a lot of that out, but when it's comes to neuro, I'm not trying to make you a spinal cord PA or anything. We're going to keep it at the level it needs to be. But neuro can be challenging because it's new stuff. There's a lot of anatomy. I mean, four-year colleges, community colleges, they just cover a very basic side of neuro, and that's all you really need. But now we're going to be providers, so we need a little more, right? And not only that, I need to help prepare you for what's coming, or you need to know that stuff. So be a little more in detail, but not to the level that you can't handle it at all. Okay, muscle tissue. When we look at muscle, we always kind of link it with nervous tissue, because those are the two types of tissue that are irritable or excitable. In other words, they can respond to electrical impulses. They can contract, extend. They're elastic, meaning they return to their resting length. So all of these describe the characteristics of muscle. When we get to nervous, we'll look at them and how they're excitable. But they're actually the ones that are generating what we call action potential. So muscles, when we look at the body as a whole, right, there's a lot of the weight. But bone is a big part of that weight. So bone, muscle, we could go into discussions about what weighs more, fat, muscle, all these things. People initially working out lose weight, and they're burning off some of that fat, but then they don't see the gains as fast. Why? Because they're starting, hopefully, to build up muscle. You start building up muscle, now your gains aren't as big, right? Because muscle weighs more. So it's a good thing. Okay. When you look at muscle and the actions, we often can think about things where, the muscle helps move us around. Right? Muscle helps to generate heat. Like you're in basic training, you're freezing at Fort Leonard Wood, Missouri, right before you're going to get sunburnt in the afternoon, and then it's going to rain that evening. In other words, I've only been there once for basic training, and I never went back. I'm never going back. That place is miserable weather. Anybody been there? I'm sure there's plenty of people have been there. Yeah, Fort Misery, right? That was, I mean, Texas has some temperature swings, but Missouri, it's extreme. When I was there, what was I saying? Okay, what we don't always think about though is muscles also control like sphincters, like your anal sphincter, keep things in the body. When you eat something, your bowels are moving, those are peristaltic contractions. Your ureters, like moving urine along, so muscles helping to move things along too. So all of those go into the characteristics of what muscle's going to do, and because of that, we have several different types. I know you've probably seen this, and you're starting cardiology, so you're spending time on cardiac muscle. I don't know how much he goes up into the differences. But if you look at just a basic chart of these, and I really like this for, I'm not saying that for a testing purpose, I'm saying it because it breaks it down where you can talk about it in one slide almost, saying, well, skeletal muscle is used for movement, your posture, you have tone. It's also muscles that we can voluntarily control. Right? It's the only type of muscle that we voluntarily control. When we look at the appearance, or when they, not that we're probably ever going to do it, looked at it under a microscope, it looks what they call striated. I'm going to show you, I know a lot of you have learned about muscle and striated, non-striated, and you just memorize that fact, right? But what's the striations in it? What gives that appearance? Well, that's the downside, not the downside to me. the good side, but it's the downside. We're going to get down to the smallest little filament in a muscle as we discuss them and show you where that comes from, why it looks like that. If you compare it to cardiac and visceral are both involuntary. We have nothing to do with controlling those for the most part. Cardiac's only going to be associated with the heart, and if you look at it under a microscope, it also appears striated. I'll come back and talk about the nucleus stuff later. When you look at visceral, you think of smooth muscle, or your viscera, or muscles that control like constriction of the pupil. Those are all types of smooth muscle, and they're going to help control things like contraction, peristalsis. Blood pressure is controlled by smooth muscle and arterial walls to an extent. There's a lot of things that control blood pressure, I understand, but we're only talking about muscle. And smooth muscle doesn't have striations because of the way that, I'm going to say this, even though you don't know probably what it is, the way the sarcomeres are lined up. I'll show you later. Alright, so when you look at the nucleus, generally cardiac and visceral muscle, and I'm not going to go into this too much even outside this. You don't even have to really think about this other than one point, is both cardiac and visceral are one central nucleus. When you look at skeletal muscle, though, it's multinucleated. If a cell is multinucleated, it's in humans, right? It's not going to go through like a mitosis and develop more of them. It just doesn't. So skeletal muscle that you have right now, you were born with. I told you this the other day. When you work out, you are going through hypertrophy. You're enlarging those individual muscle cells. You're not actually making more skeletal muscles, which I find interesting. Some don't. Right, skeletal muscle. We're going to do nomenclature first and look at how we name things. I'm going to point out the ones that you're going to confuse with each other and show you the difference. I always try to point those out, just like I did with, I see commonalities over the years, and people will miss something because of the way that they studied it and looked at it. So I start to learn and know things like you're going to confuse a melanocyte and a melanosome. Some people still study by word recognition. I'm telling you, it doesn't work in this building. Please not have to go on this floor. I'm telling you, testing to help you out stuff. But when you look at a melanosome, that is an organelle. Melanocyte is a cell. Same thing with muscle. There's a couple that sound similar, but you have to kind of know the differences to understand what you're talking about. Of course, a muscle is the entire organ. Muscles are organs as a group. And we're going to look at what kind of breaks down how we divide them up. If you look at, I'm going to start with fiber. If you say fiber, a lot of times referred to as myo referring to muscle. Right? So myofiber or a muscle fiber, that's a single muscle cell. And you got to kind of visualize this as we go through it, because everything we look at in a muscle cell, and you say that word cell, is not, what am I trying to say? When I say cell, and I say, okay, picture a cell, you picture that cell we always see, right? The generalized cell and the nucleus, and you have the rough ER, and the things you did in high school and stuff, and you see that. Muscle cell looks nothing like that, but it still has all the same components with a few additions. Right? So the pictures aren't going to look like what you visualize in your mind a lot of times as a cell. But it is still an individual cell, just a bunch of them. A myofibril is an organelle composed of filaments. We're going to get to filaments. So the two that people often confuse is myofiber, myofibril. So just like we had melanosome was an organelle inside a melanocyte, when you look at a myofibril, that's an organelle inside of a myofiber or a muscle cell. I don't want y'all to confuse them, because that shouldn't be, but if I don't talk about it like this, people will just kind of roll through them and they don't spend as much time on Okay, so inside of that organelle, the myofibril, there are filaments. And filaments are just protein molecules that consist of what they call thick and thin fibers, along with some structural and some regulatory proteins. Right? So these filaments are going to be the actual smallest unit that causes contractions. Collectively as a whole, all of these filaments, you heard of the sliding filament theory? Collectively, they're all going to help to make one big muscle contraction. Right. We're going to break down the coverings, but just for now, if you have a bundle of muscle fibers or muscle cells or a bundle of myofibers there, that is going to be called a fascicle. And so this is a fascicle. This is a fascicle. Right, so you have the entire muscle, and then you're going to have groups of fascicles. So every one of these is a fascicle, which is a collection of myofibers. If you go down to the bottom and you're working through the diagram, this is one muscle fiber or muscle cell, same thing. So you have a fascicle that is a group of muscle cells, and then inside of there we're going to have our myofibrils, which are the organelles that have the fibers. It's going to make sense, I promise you, as we go through it more. Right now we're just getting the nomenclature, but we're going to see it as we work our way down, and I'll keep repeating it so that you understand that. Generalized coverings that cover a muscle. We have the epimysium that surrounds the entire muscle. Then you're going to have, or we said inside of that entire muscle, we have fascicles. And so over here, we've kind of blown up a fascicle. And in that case, there's a perimysium surrounding it. And then the individual muscle fibers have an endomysium around it. Right, so an entire muscle, right, you have the outside, epi, you have an epimysium. Inside of that muscle, I can't do a bunch of circles. You have a bunch of circles that are fascicles. Each fascicle is going to be surrounded by a perimysium. Well, what's a fascicle? It's a group of muscle cells. Each muscle cell is going to be surrounded by an endomysium. Now, we haven't talked about neuro. Maybe you have some in cardio, but for muscles, we need to look at what's called a somatic motor unit. I don't know. I'm going to assume, just for the sake of argument, until we go through neuro next week, or not neuro, just the nervous system structure, organ, axons, dendrites of nerves and stuff. You probably don't know a lot of this, and that's fine. If you do, even better. So I'm going to try and generalize this. Inside of the spinal cord, this is a cross-section of the spinal cord. In the spinal cord, when we study neuro, we're going to see that up here in the anterior great horn, don't write that down, I'm trying to describe this to you. There are neurons that are motor neurons. And when you think of a motor neuron is going to go to a skeletal muscle. So we can control motor neurons. So out of that area, a motor neuron. Well, one motor neuron, and there are many. One motor neuron supplies a group of muscle fibers. What does that mean? That's a motor unit. Have you heard of, nowadays people are more into studying this stuff. Y'all heard of motor unit recruitment? How to increase motor unit recruitment? What we're looking at is these motor units. So every muscle, I want to say this correctly for you. Every muscle, let's just look at a bicep. If you look at a bicep, is it a collection of a lot of myofibers, muscle cells? Yes. Okay. Well, pick any, I'm going to break it down just to small numbers to help us understand. If my arm was an axon of a somatic motor neuron, meaning a neuron going to a skeletal muscle. And I'm trying not to get too much into neuro, but if you look at the end of that axon, it's going to have basically synaptic involves, right? You've heard of those? So if there's 5, let's say I'm one motor neuron and I'm reaching out to 5 people, and there's no way in IPAP or this building you're going to actually touch people. That's all right because they don't actually touch the muscle cell either. But if there's 5 of them out here, this is one somatic motor neuron and 5 different muscle cells. That's a motor unit. But there are many of them. Right? My bicep's not just 5 cells, contrary to what some of y'all are thinking as you're looking up here. Now, so I'm going to have a lot of motor units going to us. And actually, some are smaller, some are larger. Some motor units are weaker, less inefficient, and they might be 5 cells that neuron goes to. But there are a lot of other motor neurons that are going to the other cells in my bicep. So there's larger and smaller motor units. Here I just want you to understand what a basic motor unit is by definition, and it is one somatic motor neuron and all of the muscle cells it is in contact with. That's one motor unit. So, and we're gonna come back to this too. What comprises a motor unit is one somatic motor neuron and all of the muscle cells it is in contact with. Now that's the way people define it, but I was saying, kind of referencing earlier, and we'll see how much y'all are. This will give me an idea of where y'all are at with nerves and stuff. I said that I can't touch people in IPAP, right? Unless I know them very well. The somatic motor unit nerve has synaptic impulse. So are they in contact with that muscle cell? What's between them? Yeah, okay. So we understand that that's great. Synaptic cleft, a neuromuscular junction between them. So we're going to need to talk about what happens to get the signal across it. I'm getting way ahead, so because I could have just rolled through this slide and said a somatic motor unit consists of one somatic motor neuron and all of the myofibers that it stimulates. That's basic. Okay. Why am I spending so much time on it? Because that's going to help you where we're going. Right? We're going to talk about motor unit recruitment and stuff more and more. Right. When muscle develops in the skeletal side, skeletal muscle development, we're going to spend most of our time on muscle talking about skeletal muscle. At the very end of this block, we'll talk a little bit about cardiac. That's not really for this class, and smooth muscle, visceral. So the reason that I've already mentioned, when you look at skeletal muscles, and they have myoblasts, right? They're making new muscle cells during development. They start to combine together. They fuse. And once they fuse, they're unable to undergo mitosis. That's why skeletal muscle is not creating new cells. When we look at a cell, we just did that, right? We already did cellular anatomy. What is the outside of it? Well, in humans, what's the, not plants and stuff, what's the outside called? The plasma membrane. Right? Well, the plasma membrane in a muscle cell, all they do is change the beginning. Now, it's also, I should have said plasmolema, right? A plasmolema is a plasma membrane. Y'all just studied that. In muscle and nerves, they're going to change the name on you, but they're going to change the beginning and call it a sarcolema. So a sarcolema is just the plasmolema of a muscle cell. A sarcoplasm is just the cytoplasm of a muscle cell. And this is our first picture, really, where you can start to see, sorry, that this is a muscle cell. This is, all of this is one myofiber. Does that look anything close to what we saw a cell look like? That's why I referenced that earlier, so that you get an idea visually what it looks like, but all the parts are still there. You still have all the other things that a normal cell has. Another thing that names A little different is this reticulum, sarcoplasmic reticulum. Now this is very different in a muscle, so this is one to pay attention to. The sarcoplasmic reticulum in a muscle plays a large role in the function of muscles because it stores calcium. It stores almost all the calcium that's inside of that muscle cell. So when we look at a muscle cell, all of this kind of stringy orange here, all of that is basically smooth ER, or smooth endoplasmic reticulum of a muscle cell. So it's strung out kind of along there, and in certain areas, we'll get back to this, in certain areas, you're going to have like what's called a cistern, or a big storage area for that calcium. So that's the key differences there. In a muscle cell, the smooth endoplasmic reticulum is called the sarcoplasmic reticulum. It's kind of strung out all over in the orange there, and it's a storage place for calcium. Now I want you to think, this is the same slide with just the box at the bottom. I just do that so when you print on the PDF, that doesn't cover that. There's nothing else different on there. Calsequestrin is a calcium binding protein that you find inside of the sarcoplasmic reticulum. So it allows calcium to be stored in there, and you see 10,000 times higher rate. Let's picture it this way. This will help us as we go through how muscles are activated. Just for the sake of argument, we're going to say that the sarcoplasmic reticulum has this calcium binding protein, which is called calsequestrin. It sequesters calcium inside of it. But for the sake of just making it easier, let's just assume that in a muscle cell at rest, all of the calcium inside that cell is in the sarcoplasmic reticulum. Okay? Now we're making a very general statement. It's not exactly true like I like to do, but it will help your thought process. Because where we're going, if you ever study muscle, calcium is going to start a contraction when it gets dumped out of the sarcoplasmic reticulum. So rather than go through the whole facts of what calcium levels are everywhere, let's just assume that because of calsequestrin, unless a muscle, when a muscle is at rest, all the calcium is in the sarcoplasmic reticulum. And that'll do us a big favor later when we get to that. Once again, we haven't studied neuro. But when you look at that neuromuscular junction, those synaptic clefts that were out there off the end of the somatic motor neuron, and we have 5 muscle cells, when I stimulate those muscle cells at that motor unit, right? Let's just say he's just one muscle cell stimulated, that electrical potential, it's really high ion flow, but that electrical potential is going to run along the membrane of that muscle cell, right? Just like in cardio and things, it's going to depolarize the membrane basically. But in a muscle cell, we have all of these myofibrils that are kind of deep in it, all these different organelles. So we need a way for the action potential that is traveling along the sarcolemma. We need a way to stimulate the fibers that are inside of that muscle cell. The way that happens is what's called a T-tubule. And so a T-tubule goes down into the muscle cell and kind of goes to each side. So you call it invaginate in. So surrounding a muscle cell, I'm not reading those slides. The slide's the book thing. If you look at a muscle cell, and I said, okay, this muscle, let's just call it, it's a round tube. So this is one muscle cell, and it's full of myofibrils that are running all through. You have to picture that round circle there. They're all there. And so if I have an electrical potential that's going along the muscle, stimulating, that's great. But we want to be very efficient. So if we want to stimulate a muscle cell that would be in like the middle of this bottle, we're going to need to invaginate an area to get down to it. when those invaginations go down, they're still full of the interstitial fluid that surrounds that muscle. So T-tubules are a way for action potentials to travel down into the middle of the muscle fiber. Does that make sense?
So is it gap junctions that lead to those T-tubules, or is that not how it travels in cells?
No, it's a good question, but you're bringing two different things in. Because this is outside of the cell. The only place you see gap junctions is like between cardiac cells for synchronicity, because that's why I said it's a great question, because other people probably don't understand this either. Gap junctions, like in the cardiac system, allow ions to flow directly from one cell to the next without going outside. But when you need like a whiteboard, right? But when we, everybody will use a computer, it doesn't work well. I'm going to say this. When you look at like a cell, or like a neuron, an axon, the ions don't flow inside, they flow along. Remember the membrane charges are different? I'm saying this because you asked. Everybody's like, what? The charges are different. Well, actually, that's called depolarizing a membrane. So we're going to depolarize this membrane. And that means we're going to change the electrical potential, but it actually travels from one part of the membrane to the next. Right? And so it's slower than a gap junction. Because a gap junction, you look at those two cells, nothing has to go outside. You can just travel straight through them. So you can stimulate them all at the same time. Fast. That's actually an electrical connection in the body versus a, well, I don't want to choose. I could have just said, no, gap junctions aren't the same. That's why they're not the same. And you're only going to see those in cardiac, really. What was I saying? Oh, so I said y'all got it. T-tubules allow the flow of that to get down inside of there. So it's important in conducting action potentials. Action potentials, electrical flow in the body for them. Right. Another role that this T-tubule plays is that, remember I said that the sarcoplasmic reticulum stores all the calcium, and in some places the sarcoplasmic reticulum is a little more dilated. Right. Where it's going to be dilated are called terminal cisterns. So if we go to here, this is a T-tubule, right? It comes down, sorry, it goes both ways, right? It makes a T. Well, it's hard to see, and I'm going to use this one over here. On each side of the T-tubule, there's a dilated portion of the sarcoplasmic reticulum. Those are called terminal cisterns. So two terminal cisterns and one T-tubule makes what's called a triad. Right? I'm going to talk, we'll come back. When you look at it, let's look at the bottom, it'll help you understand. If this is the sarcolemma, the membrane of that muscle cell, and this is a T-tubule, these would be dilated portions of the sarcoplasmic reticulum, and they'll be flanking on each side of the T-tubule. Now we're going to switch and keep doing our nomenclature, but what that allows is if an electrical potential, and you don't have to write it on here because I'm getting way ahead of where we're going in explaining things, so you'll get it when we get there. But an electrical potential is going to travel along this membrane, and it's going to go down through this T-tubule. And the terminal cisterns that are full of calcium are sitting next to it. This is all I'm going to say, and then we'll move on. That's going to cause calcium to be released from the sarcoplasmic reticulum. Hard stop. And we're coming back to it. If I get too far ahead, or if we don't get that far today, talk about it. Hey, filaments. So a myofibril is an organelle inside of a myofiber. Right? Well, filaments are what make up a myofibril. You see how you can get real confused on these words quick? The filaments are inside of the organelle. They're I think they even say it somewhere, the myofibril is chock full of filaments. The main filaments that we're going to discuss are myosin, actin, which are contractile proteins. We work concerning ourselves with contractions, but we need to look at the structural proteins that hold this together so that it can do the contraction. So you've heard of thick and thin filaments. The thin filaments, if somebody says muscle cells and thin filaments, you're generally referencing actin, which is a contractile protein. And when you say the thick filament, you're talking about myosin, which is also a contractile protein. And so for every thick filament, there's two thin filaments. For now, just go. What are you doing? Trying to figure that out? I'll show you thick and thin in a second. Troponin and tropomyosin are regulatory proteins. We're going to go into what each one of these does, so don't fret if you're not getting it just yet. We're going to get it. We just got to build to it. Filaments inside that myofibril are arranged in compartments of what's called sarcomeres. So really, a long chain of sarcomeres is a myofibril. Look, I got confusing y'all because you're like, man, you're saying all these different things. But you got to think about analogies. The best one I like to use, we'll use the second row because it's almost full. If the second row, if this whole room is a muscle cell, what type of organelles are inside a muscle cell? Myofibrils. So we're going to say each row is a myofibril. What's inside of a myofibril is the filaments. The filament, what's that? The filaments are arranged in units called sarcomeres. And so if this is one myofibril, each one of these individuals is a sarcomere. And the sarcomeres are connected together. So I'm not going to, I was going to say, if I had them stand up and hold hands, they would be a long chain of sarcomeres, correct? But they're still called a myofibril. And they will go all the way along that muscle cell until they reach the wall. What's the wall really going to be? Sarcolemma, the cell wall. Or not cell wall, that's plants. It's plasmolemma. Right, so the last person would be touching the wall. We'll talk about what they're holding on to later. But for now, that's all we're saying is a long chain of sarcomeres is a myofibril. Each one of y'all is a sarcomere. We need to talk about individual sarcomeres, though. What's going on in that help clear up some of the nomenclature of that? Alright, because we're going to look at this crazy structure on here because this is representing sarcomeres. Right, so from here to here is one sarcomere. When you look down here at the bottom, this whole thing from here out to here is representing one sarcomere. Okay. When you look at the filaments inside of the sarcomere, they tend to overlap, depending upon how much you're contracted. So I'm just going to use 2 and 1. Remember, there's 2 thin filaments for every thick filament. So just picture them, they're kind of overlapping. I'm going to show you as we go through, but picture them being overlapped. But we've seen it's kind of tubular, right? So like I thought you were doing that. It's going to have that. It's actually not going to, we're going to talk about sliding filament theory and all this, but really you have to picture it like this, right? I thought you were doing that. Were you doing that? Going back to the previous one? I thought you were. Try to say, what are you doing? Getting ahead. You're stealing my thunder. Now. Alright. So this is a thin filament. This is a thin filament, and this is a thick filament. So for every thick filament, there's 2 thin filaments. The thick and thin filaments, when you look at them under a microscope, the myosin, the thick filament, is darker. And so this is where you get whether it looks striated or not. Is myosin and actin give this a striated appearance? in skeletal muscle, cardiac muscle, just not in visceral smooth muscle because it's set up different. Okay, we're going to slow down and get through this part because we can get this while we're in class. I love those kind of things. We can talk to them. We're going to break down a sarcomere ad nauseam. Okay? We're going to look at what are these different zones. So when they were, I always like to reference back to whoever studied this. I have to picture they were studying, they're getting very detailed. And when they got down to look at the muscle cells, and if you were doing this, just like we do everything in the military, you have an overwhelming task of looking at what's going on with something. What's the easiest way? Start breaking it down into parts and naming it different things so that you can figure out what's moving, what's going where. Well, that's exactly what they did, is they named things. They said, okay, well, by definition, and I want you to know these definitions by verbatim, because it helps you. An A band is the darker zone that they saw. So it's thick. When you define an A band, it's going to go from, you notice we'll label it from here. I'm going to draw straight through it. The A band is from here to here. Now please, if you're completely not getting this, we go through it, stop and we'll talk. I'll stay on this slide for the rest of the hour if you have to. Because this is one of the keys to understanding. When you look at an A band, let's see if it fits. I drew two lines. Let's look at that one. That's one sarcomere there on the right. Let's see. It comprises the length of thick and thin filaments with a varying overlap. Is that true? Yeah. Is there a thick filament here? Myosin? Yeah. Is there a thin filament? Yes. Are they overlapped? Yes. So that's what they defined it as. We switch to I band. Okay, an I band only has thin filaments. So let's find our I band. Right, here's I band out here. Now look, I band has an arrow out here. For the sake of trying to teach on this, I took two of the same picture and I put them side by side to show you two sarcomeres because I drew that arrow. Let's look at one thing first. Z-disc. A Z-disc is where two adjacent sarcomeres abut. Don't you like the way we phrase things in anatomy? It's where two adjacent sarcomeres are connected. Two adjacent sarcomeres are connected by the Z-disc. So in our analogy of the second row, all being sarcomeres from one end to the other, and they're holding hands, where they're holding hands, what's that called? Z-disc. Yeah. So it connects them. Okay, well, a Z-disc is also, this is the only confusing part about the I band. Is you say a Z-disc, you say, well, that's the center of an I band. So when you look at the picture, it doesn't always make sense, and that's why I went back to our analogy, is an I band is only thin filaments. Okay, let's look out here. Here's an I band, and it's only thin filaments. But there's a second part, consists of parts of two adjacent sarcomeres. I took the word abut out. They used to say we're two adjacent sarcomeres abut, but it sounded confusing for no reason. Okay, well here's another sarcomere. And here's the thin filaments, right? Here's our I band. The part I didn't take the time to do, and what I'm trying to get across to you, is if these two are next to each other, really, I'll make sure I couldn't get them close enough without losing the picture. But an I band consists of parts of two adjacent sarcomeres with a Z-disc in the middle. Does that make sense? Because people will start picturing that, because of my picture, they'll say, oh, well, here's the sarcomere with the Z-disc, and you know, they're like, what's this? This? I should just erase it out. That wouldn't exist, right? There's nothing between their hands, right? They're at the Z-disc at their hands. Tell me if you're not getting my picture. Because if not, when I ask you what an I band is, you'll get all confused and go, what do you mean it consists of parts of 2 sarcomeres? Well, it's because an I band has a Z disc in the middle. It's where they're connected, yes.
So you're saying where it says the 2 Z disc spots, that is the same.
Yeah. Yeah, so if I had, like, stick your, like you're holding hands, you don't have to hold hands, that's right. This would be the Z disc. And up to, if you want to say it this way, where does an I band really start in that sarcomere from? At the end of that thick filament. You see that up there, the red, the dark red? So from one dark red in this sarcomere to the next one is your I band, right? And so their arms would be the I band. From here to here, and then your thick filaments would be out there. Is that, y'all kind of seeing that? Because there's only one Z disc there.
What you just said was really good.
No, that works.
There's only one Z disc.
Yeah, there's only one Z disc. That's all you need. You know, unless one of these people in this row has 2 hands on 1 arm. That would really throw us off.
So what's with that though? Because in between 2 of those I band, we see a tightened filament. Where does that come into play? That spider looking like slinky looking thing. Yeah.
We're getting there.
Oh.
We're getting there.
So that is also a part of I band though, is it? Or is it not?
Yes. Yes, it is there. It's not defined that way, but tighten is there. Actually, we're going to see that what you can't see is tighten goes all the way back to the M line. You see what I mean? So that's why they don't say that's just, it is part of it though. You are correct. We're going to get to tighten. Okay, I think we got it. Now the H zone. The H zone is the center of an A band and only contains thick filaments. Is that true? Let's see. Let's draw some lines. Here's your H zone. Is there any yellow actin filaments in that area? No. So the H zone, by definition, only contains thick filaments, and it's the center of an A band. Right? I didn't draw that, but I remember here's our A band. Now that I've drawn all of it. So an A band consists of thick and thin filaments, correct? But the H zone is a portion of that that only contains thick filaments. They were defined, remember, go back to people who were looking at this and trying to define out, to organize it. And then you throw in the M line. So the M line is actually a myomesin molecule. The M line is the center of the sarcomere, or the center of the A band. Right? I know H zone is the center. In other words, you can't ask it that way. Which is the center of an A band, the H zone or the M line? Well, by definition, the H zone is. But is the M in the middle? Yeah. So you can't ask it that way. For those of you who just thought that. How are we feeling about it? If you're thinking it doesn't seem as complicated as I thought, then we're in a good spot. Now you probably still don't understand it, but we're getting there. If you got at least that thought. Now the concept that we're learning is, what they wanted to know, I'm sure, is how does a muscle contract? So they got it down to the smallest unit. And what we're going to see is the sliding filament theory. They're going to slide together. And so we're going to define what's actually moving. What's causing that movement. That's why it's key to understanding how it's set up. Because, she was really in a rush to get you tightened. Now we're here. No, I'm just kidding. It was a great question. It was just a little early. When you break down the proteins inside of the muscle, right, particularly down to the filaments, really what they are is proteins. Of course, everything's protein, right? So there are contractile proteins. The thick and thin filaments, actin and myosin, are contractile proteins. They are technically going to contract or slide together. We're going to see that. The other proteins are going to help with either starting or ending the muscle contraction or holding it together while it does that. So you have regulatory proteins. Regulatory proteins are what's going to determine whether we have a contraction or not. They're going to regulate the contraction. Structural proteins are going to hold this muscle cell, these fibers together while they do their job. And that includes titin, myomesin, dystrophin, and then what they call sarcolemmal proteins. As you'll see with a lot of things we talk about, what could we also, just in basic cellular anatomy, could we call a sarcolemmal protein? You know, I like to refer back to this all the time. It's an integral protein inside the membrane. So those won't be hard. Alright, so learn them that way. That is definitely something I would ask you. Which one of these is a regulatory protein? Even though I feel like when we go through it, it would be a very obvious question if I did ask that, because we're going to look at each one individually. Here's myomesin. Or sorry, myomesin. Here's myosin. Myosin is set up to where half points one way and half points the other. So this is just a thick filament. We pulled it out of there and looked at it. What would be here in the center really if we looked at the whole section? Look at that M line. I think I heard somebody say it. Right, which is that's where myomesin comes in. That's a myomesin molecule. And so the thick filament faces each way. So you have tails and the heads of this myosin molecule. If you think of it, it kind of looks like a golf club, right? Golf club with the tails all wound together. And so what we have here is a bunch of them, right? I'm going to erase it. So slow. Okay. So it looks like this where the tails are wound together in the head, but there's a bunch of them. A whole lot of them. And then the other half go this way. Okay, so that's what myosin looks like. When you look at why myosin is important, is the myosin heads have the ability to form what's called a cross bridge. We're going to build upon this, but for now, the heads of myosin are called cross bridges. Okay, we're going to look at how muscle moves. That's going to be a key to that. And the way it's going to do that, without bringing don't bring biochem in here. We're not going to talk about how phosphate groups move and stuff in here. You know, I don't like that. Hope some of y'all are with me. But we do need to understand basics. And if you don't ever understand in anatomy 1, I know this sounds negative, what bind and hydrolyze actually means, I'm not real worried about that. That's not my job. My job is to show you how this muscle cell functions. But you have to have the ability to bind and hydrolyze ATP, which is basically just moving phosphate groups around, creating energy, right? We're going to change the shape. We're going to move towards or away from that midline. And then myosin must also bind reversibly to actin. So all three of these are what myosin needs to do. Let's look at, let's just finish the contractile proteins here. I'll give you a break. This is actin. Actin looks like a string of pearls. So the thin filaments that we saw that were yellow, each one of these is an actin molecule. Strung together. So it looks a little different than myosin. But each one of those, I get distracted, yeah. We're going to do this slide and the next one and we'll take a break. Each actin molecule has a myosin binding site. Okay, when you look at different molecules, it's always important to look at like, well, which one has the binding site? Right? Which one has this? Because truthfully, the myosin head is going to bind to actin. Where is it going to bind? The myosin binding site. Okay, well, you noticed already on a lot of tests, you know, we get specific. So which side has the myosin binding site? Actin, because actin's waiting for myosin to bind to it. How do I know that y'all messed that up? Because I've asked that before and people go, Well, isn't there a binding site on the myosin head? I'm with you. I'm a redneck from East Texas. I get it. I get that thought process. But is it that way in the books? No. The binding site's on active. Now, biochem could go a lot more. I'm sorry, I'm not supposed to say that on the day. I don't know. I try not to bring up any other classes on the day that you took their exam. Because it doesn't always go away. You'll shut down listening to me. Okay. So what we need to bring back in then is myosin heads will bind reversibly to actin at the myosin binding site. So each one of those black dots is a myosin binding site. We have regulatory proteins that prevent that from happening or let it happen. The regulatory proteins, troponin and tropomyosin, they often call it the troponin tropomyosin complex. They're going to cover the binding sites on actin. Right? So you have tropomyosin here that's brown, and then these little blue ones are troponin. And you notice now we can't see the little black dots. So troponin and tropomyosin in a muscle cell at rest are covering the binding sites. They're blocking it. Last thing before the break. So, in order to start a contraction, or say it another way, in order for myosin to be able to bind to actin, troponin and tropomyosin need to get out of the way. Because they're physically blocking, like a receptor blocker, right? In the cardiac cells. So movement of the troponin tropomyosin complex allows the contraction to begin. I'm going to finish on this statement. We'll take a break unless you got a question. When we take calcium out of the sarcoplasmic reticulum in a muscle cell and put it into the myoplasma of that cell, calcium is going to bind to troponin and tell these two to get out of the way. That's what we're going to look at next. All right, let's do it. A little too animated.
But I also yeah, that's true. Like that. But it also this weekend? Yeah, that's a lot of people. Yeah, but that's not the problem though. Yeah, you don't want to go. Yeah, family's there. Go somewhere just away from them. Are they still there? No.
So they do like a little bit.
So
Yeah. Here you go. You're still in your trap.
how did you? Start getting patient.
Have you seen that with your hair, with your wife?
Yeah.
Good question. Yeah, no, you can shape. So like, depending what version you have, the one that you did last week, that's an updated.
I do lecture for an hour.
Yeah. Usually we try to get first argument. I don't know how long you expect to stay in the lab. If the whole time. Do you feel like if we have yeah, time is only like
Where's it at on here?
I'll
library.
Every Thursday we did updates, so my guy, it's a bit cardiac because of the bigger deck, but it's AC and ABC now. I live yeah, mine did too. I just started off. Which is why they tell you like that's really good. I don't care. I'm a fan of this.
I like my
My wife has one that doesn't great for me. All the bottles are like.
I'm so focused on the exam.
Alright. Let's get it going. Somebody had a good question at the break. Somebody had a good question that I felt that I didn't mention to y'all. Is what did we call? We talked about the sarcoplasmic reticulum. We said it's smooth, and they keyed in like appropriate. So we go. Well, if they're smooth, is there not roughy in a muscle cell? If so, why didn't you talk about it? Kind of a given in my mind to think that way, but it's not. That's why it's a good question. I don't really talk about the rough ER in the muscle cell. Is it there? Yes. What did we just talk about last hour? What officially are myosin, actin, proteins? Where do we make proteins? Rough ER, right? Go to the Golgi body and all that. So we covered that in the general cell. That's occurring here too. We're just not going into it. And you're going to see that's pretty common. When we get into the neuron, we're not going to talk about that stuff either, really. In a sense, there are going to be parts that we leave out in the general cell, so we can focus on what's different, like with a muscle cell versus a general cell, or what's different with a neuron versus. Okay. So that's why I didn't bring them up. Made sense, I hope. Yeah. All right. Now let's get into what this. Well, we got to finish the molecules, and then we'll tie together what's going on. When you look at a sarcomere, somebody asked about titin earlier, and it's helping to hold things together. Well, what titin does is titin spans half of a sarcomere. So it's going to go from a Z disc, and it's this coil piece here, that's elastic. It's going to go all the way down, but if you notice, it's not stopping here. It goes all the way to the M line. And the same thing's going to happen over here. And so I often think of titin like a little spring. As the muscle contract, almost like, actually now that I think about it, titin's like the IUD I was handling the other day. The compression of the springs. And as they slide in, these actin myosin slide in, titin helps to return them to their resting line. It's like a spring. So it's called a regulatory protein. It's helping to regulate the contraction. Or hold it together. Sorry. Erase that. The structural protein that is helping to hold this all together. Troponin and tropomyosin are the only regulatory proteins there. Dystrophin. We did an analogy earlier, and everybody held hands, and I said, I left it at that point and said, the people at the end of the second row there were their individual sarcomere themselves, and they're, like, she would be connected to the sarcomere next to her by a Z disc, but she's at the wall of the plasmine or sarcoline, right? Her hand would be touching nothing, right? Or it would be touching the plasmine, but really it's not touching. I'm gonna walk all the way out there just for the effect. She's not even standing up for me in the room. I'm just kidding. I'm just kidding. You're taking it. Her hand's here, right against the wall. But just like in other areas, it's not actually touching that membrane. So what are the sarcomeres gonna do? Like y'all were doing earlier. They're all gonna pull, and you would see that whole row come closer together, right? And so this is pulling on the plasmoline. Keep on it. It is with the sarcoline, right? So that's a lot of force from that one line of sarcomeres. And so sitting between like the Z disc of this sarcomere and the sarcolemma is dystrophin. This is another structural protein. And what dystrophin does is it helps distribute the force of all these sarcomeres contracting in. It's going to pull. Just like they were going to try and pull the walls down here on it. And there'd be like a connection between those last people and the sarcomere. Yes?
Can you do it over here? Just
No.
I'm just kidding.
I can see the look on your face when you ask. Oh, that's with it. Okay. Have we heard of dystrophin before? Have we heard of issues with dystrophin problems? Do we know who Jerry Lewis is? You know? Didn't he do muscular dystrophy? We've heard muscular dystrophy. Muscular dystrophy is a problem with dystrophin. Either it's abnormal, because absent dystrophin here, what they found is these contractions over time will start to tear the sarcolemma. And ultimately, you'll damage that muscle cell. But if it's going on in all your muscles, you can see how that's going to lead very quickly to being very debilitating. Just say, muscular dystrophy. That's the problem. I got a slide on that later, but that's what dystrophin does. Myomesin, the third structural protein here, is we find myomesin at the M1. That's all I'm going to say about it. Myomesin is the middle of that sarcomere there. And then sarcolemmal proteins are just integral membrane proteins. So if we define it out, we would say, well, here's the last person in the second row over there, and here's the sarcolemma, and here's dystrophin that connects them, and then you need this sarcolemmal protein for dystrophin to attach to this. That's all it's doing. So it becomes just a definition thing. Which ones of these are structural proteins? Which ones are regulatory? Yeah.
So is dystrophin almost like a damper?
Yes. Yeah. I would agree. Like a damper, a resistor, almost like dystrophin could, be tight. It could kind of stretch a little. A shock. I was doing those the other day. If you don't know this, your shocks. People think that your shocks when you go down in the car, but really your shocks catch your car when it bounces up. Keep it from doing that when going down. Does that make sense? Which is a damper. So it's just, that's a great analogy. In my mind, some of y'all are like, what the hell are y'all talking about? I already got it. It's a structural protein. That's all I see. Alright, so we get to tie all that together and say contraction and relaxation of these skeletal muscle fibers. So you have to bring some stuff back up. So every myofiber, or each muscle cell, is innervated by one somatic motor neuron. It goes all the way back to the beginning that we covered last hour, that a motor unit is one somatic motor neuron and all of the individual muscle cells that it's in contact with. But there are many, many motor units. There are many, many muscle cells. And where they meet up with the nerve to the muscle, that's your neuromuscular junction. So we haven't discussed action potentials, but for now, in order to get a muscle to contract, you need to depolarize sarcolemma, the plasma membrane of that muscle cell. Once you depolarize it, that will cause an action potential that flows along that membrane. In other words, we need to stimulate the muscle cell. Right? That's the signal that it's time. And so as we stimulate one muscle cell, there's going to be an action potential that runs along the sarcolemma, down through the T tubules, and depolarizes that whole membrane along as it goes. Right? Just for now, think of, if you don't know what an action potential is, think of an action potential as electrical flow in the body, the current of the body. What is the current of the body? Ion flow. There's an exchange of ions going on that we're not talking about yet. You look at power lines, that's a flow of electrons. different, it's just electrons instead. Okay, so where that somatic motor neuron's synaptic impulse, the end of that, is close to that muscle cell it's going to stimulate, you get what's called the neuromuscular junction. And I know you get this in multiple classes this semester, but so this is this one synaptic impulse of a neuron. Right? And so you see a neuron here with lots of them coming down. And so, but we're looking at how it gets to one cell. And so if you look at this part, and then you come over here and blow it up a little bit, that's the end. So what's actually happening is there are neurotransmitter that is in that synaptic impulse. Now all skeletal muscles, all of the neurons that innervate skeletal muscles release acetylcholine. And here's one of the first facts for the rest of your life while you test on PA subjects, or any, actually even in biochem. If you're discussing a question about neurotransmitters, this is a hard, fast rule. I'm helping you out here. If you look at a question about, and you have a bunch of lists of neurotransmitters as your answers, and it gives you the definition of what it does, and you don't know what, you read it, and you're like, I'm not sure what that is. When you look at the list, you're going to have 5 answer choices in biochem, and wherever, on the pens. If you don't, absolutely, you just don't know the answer, you must answer acetylcholine. You must. If not, you're going to get it wrong. And I'll give you the stats on it. I guarantee you that if you were to sit down on a neurotransmitter test and answer acetylcholine every time it was an answer choice, you would get 80 to 90% of those questions right. Because acetylcholine is the most used neurotransmitter in the body. So generally, the answer to a neurotransmitter is going to be acetylcholine. Except for other ones we're going to talk about. But remember what I said, if you're reading it and you absolutely have no clue, I don't know what any of these are, just remember acetylcholine. Tell me. You're going to see it pay off later. You're going to be like, I don't know. Did we even talk about that? Just answer acetylcholine if you don't know. You'll be right. You'll probably be right. But all skeletal muscles use acetylcholine. They don't use any other neurotransmitter. And what it does is it's going to release, we'll get to how it releases stuff later. You're going to, basically exocytosis is going to release a neurotransmitter into the space between the neuron and the muscle called the synaptic cleft, but it's also called the neuromuscular junction, neuron to muscle in this case. And when it releases acetylcholine out there, it's going to move across the synaptic cleft, and it's looking for some acetylcholine receptors on what's called the motor end plate. So the muscle, the portion of the muscle cell here, where that synaptic cleft is, this bottom portion is called the motor end plate. And that's where we're going to see that there are receptors, or integral proteins, right? Receptors are a type of integral protein there for acetylcholine. Right? And so when acetylcholine crosses that synaptic cleft, it's going to bind with acetylcholine receptors. Specifically, 2 acetylcholines are going to bind with that receptor. When that happens, that will open a channel to allow mainly sodium. We're going to discuss sodium a lot. A cation, a positive ion, it's going to allow it to now go into this muscle cell, right, which triggers an action potential. Some of this makes it.
To cancel, you can also press the digital crown on your watch. Okay, cancel.
What was it sending? You know, they're probably just calling boss up or something. No, sorry about that. So you have, the hardest part is that we haven't covered a lot of neuro and keeping people on track without overwhelming it. But so I'm trying to generalize here. You have a synaptic embol that releases acetylcholine, right, into the synaptic cleft. Acetylcholine will cross the synaptic cleft, where it's going to bind with, I've been ignoring this screen all day. Now that you said that, I realize it. It's going to cross the synaptic cleft, bind with acetylcholine. That's going to open a channel for basically sodium to flow into that cell. If we had studied neuro, you'd already know that when sodium movement, like voltage-gated sodium channels, open and allow sodium to come in, or these receptor channels, that starts to depolarize the membrane of that muscle cell. Right? Depolarizing it is how we're going to move the electricity along. It's how we're going to stimulate it. We're being very generic, but I don't want to get too far ahead of it as we go. Okay? So you could say an action potential will travel down the neuron, stimulate, right? You'll have exocytosis of that acetylcholine, and your goal is to take the action potential that's in a neuron, because the neuron's electrically excitable. You're going to take that action potential and you have to cross the synaptic cleft and stimulate the next muscle. In order to stimulate the muscle cell on the other side. Because we have to cross the cleft, we're going to use a neurotransmitter to do that. In the heart, it doesn't occur. The heart contracts all at once, right? Because of your gap junctions. Now I'm getting way out there, but I'm trying to link some things in. Sometimes you get too far out there without, and don't go into the info, you just confuse people more. So let's just keep going. We are definitely coming back. Alright. So what we've backed up and we're looking at, excitation, contraction, coupling, means how are we going to start causing contractions? So we have to make an assumption that there is an action potential sweeping along the sarcolemma, going down through the T tubules and just continuing across this muscle. So on this side, that action potential hasn't started yet, so we're at relaxation. So when the muscles get relaxed, what was the thing I said? We're just going to assume that all the calcium is where? The sarcoplasmic reticulum, because of calciquestion. So in that left side of the picture, all of the calcium is in the sarcoplasmic reticulum. That means that the sarcomeres are at rest. They're not contracting. Alright, and that means also that troponin and tropomyosin are covering the binding sites on actin. All of that's true. In order to get this muscle to contract, we need to move troponin and tropomyosin out of the way. Remember, they regulate it. So in the right side, what's happening is that action potential, I shouldn't have drawn it on the left one. That action potential is going to travel along the sarcolemma, down through the T tubules, all the way along, and it's depolarizing the membrane as it goes. When that happens, that will depolarize the membrane of the sarcoplasmic reticulum. Remember, there's one on each side. One T tubule and two terminal cisterns is called a triad. And y'all are attractive today. Y'all are a smart class. Right? You're amazed how many times I do that stuff in other classes where they come along and it's just dead silent. Dead silent. Even though half of them know. Anyway. So depolarization of the sarcolemma causes depolarization of the membrane of the sarcoplasmic reticulum, which is full of calcium. So in that case, that will cause the release of calcium into the myoplasm, right, or sarcoplasm, out into here. So now we have calcium outside of the sarcoplasmic reticulum. What that calcium will do immediately is it will bind with troponin. So when you bind calcium to troponin, that move, I know I wrote changes the shape of troponin. I don't care about changes the shape. Here's the key. When calcium is present inside of the rest of that cell, calcium will immediately bind with troponin and that will cause troponin and tropomyosin to move out of the way and uncover the binding sites on actin. Okay? They're going to slide out of the way, so that if we look at the picture on the left, you notice you have tropomyosin and the blue ones being troponin. Both of those are covering those dots, the binding sites. Myosin head binding sites there. And in this picture, we can see that they're uncovered. And in that case, the myosin head can now bind or form a cross bridge with actin. Okay? So calcium causes troponin and tropomyosin to move out of the way, and now the myosin head will bind to actin at that binding site. That's the start of the contraction. That's the start of what we call the sliding filament theory. So once you expose the myosin binding sites, myosin heads will immediately form a cross bridge or bind to actin. And what that's going to do, we're about to go through a bunch of funky pictures, but if we picture I'm the M line straight up and down, and each arm is one myosin molecule on each side, right? You got the myosin heads, and I can't draw enough though, but on top of each of my arms would be the actin, the thin filaments, because this is the thick filament. And what they're going to do is they're going to come up and bind to actin and rotate towards the center and kind of pull that in. Now, I'm going to show you on the next few pictures a better than my analogy here. We're going to rotate the myosin heads. You see what I mean? Because before I got there, I had to throw this picture out there. Alright, we're going to discuss the sliding filament theory. And while we do this, we're going to go back to our zones, our H zone, our I bands, A bands, and we're going to see another reason why they define them out is we're going to watch what happens when a sarcomere shortens. Right, so in the top picture here, I'm going to spend a little time here on this, and then we'll move on. We'll come back to the same picture. We can see all of our zones. We have, actually, this is a good picture to show you. Oh yeah, I need to change. I never even realized that I had all this time. It's a better picture than what I had on the first one. You have two sarcomeres. Right? This, when you look at it, this is one sarcomere. Here's another sarcomere over here. Here's a Z disc. So that makes a good, better picture of what I was talking about at the I band, right? I need to switch that. Somebody make a note. Instead of mine. Right, so what we want to pay attention to now, though, is they're going to slide. If you look at the thin filaments and the thick filaments, what we're going to do is see them slide together. And as they do that, we're going to see that the zones, some of the zones, are going to get smaller, particularly the H and the I. The H zone and the I band will change their length. Let's leave it at that, and I'll come back to this picture, and we'll look at the myograph side of that. So this is the sliding filament theory. Is if we start, just write out your book, the myosin heads hydrolyze ATP and become orientated. All right, so I didn't cover that yet. When are muscles at rest? The myosin heads are just sitting out there, right? The binding sites are covered on actin and all, so they're just sitting there. Now this never occurred. They would be flaccid if this occurred all the time. Just be an example. And so they're ready. The myosin heads have hydrolyzed. What hydrolyzed means in this case is they're energized. They're ready. So myosin, right, is like my dog at the window barking at a deer like it's nothing. I don't know why I thought of that. You know, he's just charged up and ready. If I open the door, he can go after the deer. You know, he's barking. I don't understand dogs. You don't have wild dogs. Your dogs bark when people pull up. Crazy dog. I'm like, shut up. Nobody's coming. We'll shoot them if they come. Nobody's coming. Okay. So you got myosin heads out here. And I'm shaking because they're energized and ready, but they need troponin and tropomyosin to get out of the way. But troponin and tropomyosin are not going to get out of the way unless you have calcium present. So it's just sitting there energized, which means that the myosin heads have hydrolyzed ATP. Even if you don't know what hydrolyzed means. They're energized, they're ready. And two, the myosin heads bind. Okay, so that means we dumped calcium into that muscle cell. Alright, so calcium came in here and moved troponin and tropomyosin out of the way. In that case, myosin heads will come up and bind reversibly to actin. Reversibly being the key word. And then they will complete what's called a power stroke. Right? Remember, we're talking about sliding in, one thick filament, too thin. Basically, the thick filament, I'll show you on the next picture, they're pulling the thin filaments towards the end line. Right, the thick filament, I'm the thick filament, and here's our myosin molecules going each way from the M line, and on the top and bottom of this, I would have the thin filaments, right? So what the myosin heads are going to do is come up and bind to actin, and then they're going to do a power stroke, which is where the head of that molecule rotates towards the center, and then it will release. Now remember, we have billions of these, so it's not just one. And so, but each actin molecule has a binding site for a myosin head, and that myosin head is going to come up, attach to it reversibly, which is called binding, and it will rotate towards the center, then release. So it's kind of going to pull it in. I got a fancy graphic coming up too that'll better than my fingers even. Okay, that's the basics. But we need myosin to release. Okay? So myosin's already hydrolyzed and ready. Once troponin moves out of the way, it will come up and attach, and I said it's reversibly. What it takes from myosin does its power stroke, and then it needs to release so it can go back and get ready to do another one. The only way that myosin can release from actin is by binding ATP again. Right? So it'll bind ATP again. That will cause the release of the myosin heads from actin. And then guess what the myosin heads will immediately do again? Hydrolyze ATP. They will energize themselves again, immediately. Now don't bring biochem in here, because you're taking biochem, and I said it twice today, I'm not supposed to, but I have to. In biochem, you would start going, okay, because you'll go in my class and you'll go, all right, so what did he say was key? Calcium's required to move troponin out of the way. The myosin heads then have hydrolyzed ATP, and they can bind to the actin, myosin binding site on actin, and then they're going to do a power stroke. And in order to release, they're going to bind ATP again, and then they will immediately hydrolyze ATP. Hydrolyze, re-energize. They're energized, use energy, but what you'll start bringing in is like, okay, so he said that they hydrolyze. If you get into a study group for my classes, somebody comes in and goes, okay, he said it hydrolyzed ATP already, and then he said in order to release, it had to bind. So when it binds, this phosphate group is going to do that, shut them down right then, and say, this is not biochem. No, I'm kidding, but because it confuses you on my stuff. Not that it's that hard to understand, hydrolyze and bind, but it's just fun to talk about. I don't like biochem. I have nothing against Johnson and them. I tell them that's one of those things. I will say it to your face. I don't know. I got something against biochem. It was my nemesis when I was here. Okay. So going back and looking at these fancy contractions, we want to know, and I want you to know, which bands will decrease. In other words, when a sarcomere shortens, what zones shorten what don't? You have three different examples here: relaxed, partially contracted, fully contracted. In this case, let me know if you don't get it as we're going through it. The H and the I zone Our I band decrease. Okay, let's look at the H zone. This is relaxed. Here's your H zone. Remember, the H zone is the center of an A band and only includes thick filaments. That's why you got to know those definitions. Once this sarcomere starts to contract, the H zone will get smaller. Alright? So what is happening here to the H zone? I want to describe it a little differently. You can either learn that the H and the I zones get smaller, or you can figure out visually why they're doing that. Remember I tried to say just a second ago that the thick filament kind of just sits here and pulls actin in on it? And the reason I said that is that when it's pulling it in, look here. Another way you can define it, you see the actin molecules are that far apart? And how far apart are they down here? They're closer. That's because, remember, the H zone only includes thick filaments, and so you still define the H zone as only thick filaments, but what happens is you've shortened it by pulling actin towards the center. Please let me know if that doesn't make sense. That's why the H zone shortens. And well, what about, and then you can go down to a fully contracted and it kind of disappeared there. Let's look at the I band. The I band, why would it get smaller when this sarcomere contracts, or sarcomeres? Here's your I band at rest, and now down here is, I like to use big screen, I just can't get it. Down here it's going to get smaller. And why is that? Well, remember, actin's being pulled towards the center, and actin's being pulled towards the center. And if the definition of an I band is where two adjacent sarcomeres connect and a Z disc in the middle, then you kind of see how that's going to change its length there. And the A band will never change, because I said think of the myosin molecule as staying stationary. That helps me when I think about it. It's actin that's moving and myosin is pulling. Here's another example, and I really like to use and look at the micrographs here because you can kind of see the shading where here's a Z disc, here's a Z disc, so that's a one sarcomere, here's your inline, so this would make this your H zone, so it only has those thick filaments, but here's your A band in total, so you have some overlap now because an A band consists of both. And if we were to follow this down in this picture, we can see very clearly that A band doesn't change lengths. But the I band, okay, if you look here, it's getting, you look here and here, it's getting closer together, and then here it's even closer. Okay. Same thing with the H zone. The H zone started out really big up here, and now it's smaller. And then it's almost non-existent here. This is fully contracted. So after all of that, you get one of my high-speed graphics that will stay, even if we go fully 4D, 5D, 8D, if that exists in graphics, I will still keep this one. It's high speed. I mean, it just explains it all. If you stare at this long enough, you'll see zebras. I mean, no, you'll understand. I want you to watch the H and the I. See, the A is staying the same, H and I, and now you can kind of see red is active. Purple or blue, whatever it is up there, that's your myosin. And if you look at the end line where it looks like little diamonds or dots, depending on how close you are to it, it'll cycle to it again. And you can watch them kind of slide together. Now, if you do start this, while you're waiting for it to load, if you do start to see zebras this weekend looking at it, it's Memorial Day and you've been drinking too much. You need to stop studying if you've been drinking and sober up then study. But it takes a while to load. I had to say something. Now, when I look at it, that's a great graphic. I know y'all, how is this? simple graphics to me are better.
Just want to make sure I get this right. So the H and I, they shorten just because of the cross bridge that is contracted?
Yeah. Yeah, they shorten because the myosin heads are pulling acting towards the center. Did y'all hear that? Yes, you know. You said it like you already had it. That's right. You got it. Somebody else probably confusing here, they're just not asking. They're like, I'm not worried about this till next week. Then they're gonna come back, and they're gonna see this graphic, and they didn't listen today. They're gonna be griping going, dude's got a terrible graphic. What is this supposed to be telling? Just write zebras up there. That'll keep your mind remember the whole time. The way the memory works. All right. Animation. Maybe time is animation. That's good. Well, that's what they say. I haven't digressed hardly at all today. You know, they say you watch all these videos now, and they don't think we went to the moon. They're talking about how good the video quality was compared to the actual like video games and stuff of that day and age. I don't know. Can you see the flag on the moon with the telescope? High power? I don't know. Anybody know? I think you can. See, a good conspiracy theory will always have a follow-up label. And if all else fails, you just say, well, I don't trust the government. I'm not saying I do. Not saying I do, but didn't go to the moon. That would be something if we did. Okay, rigor mortis. I'm not saying I think we did. How could Armstrong, we brought him up yesterday, right? How could Armstrong lie for his whole life? The guy was pretty convincing. That's weird how that, we went full circle. You circled back. All right, rigor mortis. Rigor mortis is very interesting because after you die, we all know that rigor mortis sets in. But really it happens about 3 to 4 hours after you're dead because you're no longer maintaining the membranes. Each cell has organelles inside it. We're talking about muscle cells. Muscle cells have a sarcoplasmic reticulum. Well, they're membrane bound. Remember, almost everything in a cell membrane bound. So if your cell membranes start to break down, so is your sarcoplasmic reticulum. It will release all that calcium out into all your muscles at the same time, and you are going to full rigor mortis. One big body contraction, all together. In other words, when calcium dumps out in the cell, all of the binding sites will be uncovered immediately. And every bit of myosin that was waiting, right, will go up and form a cross bridge. Okay? That's going to happen within a couple hours. The membranes will break down, all the calcium gets dumped out there. Picture every one of your myosin heads doing one power stroke at the same time, and you're fully maximally contracted in all your muscles. That is rigor mortis. Now, what's the problem? Why are we going to stay in rigor mortis? The answer is, what does it take for the myosin head to release from active? ATP. You have to bind ATP again, but you're dead. You're no longer making. When you die, you stop making ATP. Right? The Krebs cycle gets shut down. The factory is done. So you can't make ATP anymore. So because of that, the cross bridges from the myosin can always just say myosin head attaching to actin. It's called a cross bridge. So those myosin heads cannot be attached to actin. And so within about 24 hours after death, though, they can actually judge how long you've been dead by rigor mortis in a sense too, because after about 24 hours, these proteolytic enzymes come in and start to digest, well, not only the whole body, but start to digest the connections between those cross bridges. They'll digest them, and the body will flaccid, relax again. So if you're going to move the body, I suggest within the first 24 hours, because a rigid body is much easier to carry than one that's just, it weighs twice as much when it's limp like that.
So we should seize the moment.
Seize them up, we gotta get rid of them quick. Right? You're gonna be doing like a dead man's fireman's carry otherwise. The other one, you can probably just step out and against the wall. Picturing that. Yeah, weekend at Bernie's, exactly. Yeah, and that's where I live too, Bernie. Okay. Yeah, I'm gonna stop. We're just gonna keep rolling in more muscle stuff. There's no real cool stopping point coming. So that's gonna be it for the day. I think we covered it out. Hopefully you got something out of it. Hopefully I wasn't too abstract about the way I explain things. This is one of those areas, I say this a lot about certain areas. If you really are having trouble with sarcomere and its movement, we just need to talk separately. Because I often, when you ask me things like, well, I go, no, you're thinking of this now. It's hard to do with 67 people who aren't really asking you questions and stuff. If this really seems complex to you, next week, come and get with me. I'll sit with you on a whiteboard. We'll go over it in less than a couple minutes. You'll be like, why didn't you just say that in class? I'm like, I did. I just didn't say it the way you wanted to hear it. All right, y'all, hey, be safe. I know I'm not the safety person. I'm dealing with